My name's Sarah Birchall. I'm based here in Oxford in the UK, and I'm going to show you how to extract DNA from fruit. So good fruits to extract DNA from are A, nice and squishy because it makes your job much easier, and B, they should be something known as polyploid. So in each of the cells in the human body, or most of them anyway, there are 2 copies of each chromosome. So we say they are diploid. In something like a banana, there are 3 copies per cell, so that is triploid. In a kiwi fruit, they are hexaploid, that means there's 6 copies. And in a strawberry, they are octaploid, which means they have 8 copies of each chromosome per cell, which means they give us a nice big high yield of DNA, so it makes it easier to see it when we actually extract it. So to do this, I have taken one lovely yummy strawberry like this, take the leaves off the top, and then crush it mercilessly with a fork until you've got something that looks a little bit like a smoothie. And then you're going to need to add to it something called an extraction buffer. And I've made that in this jug here. This is 100 millilitres of ordinary tap water, and I've added to it a teaspoon of washing up liquid and a teaspoon of salt. So this is our extraction buffer. And we need to add 2 tablespoons of this into our strawberry mixture. Now you need to mash it around with your fork again, give it a good stir and mash it nicely, but very carefully because you don't want to make a load of bubbles. Remember, you've just added soapy liquid to this, so it's very easy to make a lot of bubbles. Now the plan is to keep mashing and mashing for about a minute. And what we're doing here is breaking open more of the strawberry cells. And when we break open the cells, hopefully we'll get a release of DNA into the liquid. So the soap that's in our extraction buffer actually helps to break down the lipid wall inside the cell. So it helps to release the DNA into the liquid. So that's our first ingredient. And then the salt that we added as well, that's going to help the DNA to clump together, which will make it easier for us to see at the end. So now I've got a beautiful strawberry sort of soapy liquid that you definitely wouldn't want to drink. Then I'm going to filter that. So I've got here an old spice jar that I've washed out, and I've got a sieve with a piece of kitchen roll in it. And I'm hopefully going to catch the liquid as it comes out of here. There you go, it's coming through nicely without any spillage at all. That was unexpected. And that's now dripping down into my jar. So hopefully most of the strawberry will be left behind in the sieve at the top. And when it's coming towards the end, you can very gently give it a squeeze just to speed things up. Be really, really careful so that you don't break open your tissue and release lots of cells into your mix. That'll do. Just a little bit more. Look at that, beautiful. So now I have this lovely pink strawberry mix which contains the contents of the cells and lots of my lovely DNA, hopefully. The other thing I did to prepare for this was to take some laboratory alcohol and put it in the freezer overnight, so it's really nice and cold. You don't have to use laboratory alcohol at home, obviously. A nice clear spirit like vodka would work nicely, and you can even try it using surgical spirit if you have that, because that's mostly ethanol or alcohol anyway. So with our ethanol here, what we need to do is tip it very gently down the side of the jar, because we are hoping to get it to sit on top of our strawberry mixture. There's a little bit going there, help! But as you can see, that's about 30 millilitres from that pot there. And as you can see, I've made 2 distinct layers. We've got the strawberry on the bottom, and we've got the alcohol is sitting on the top because it's less dense. And then what you can see happening, if I hold it really super close for you, can you see that? Look at that. Just at the point where the 2 layers mix, you start to get strands appearing. And I'm just using a kebab skewer here because I happen to have one handy. And you can literally pull it out, and that is DNA. Now it's not going to be pure strawberry DNA, there's going to be little bits of cell contents in there as well. And also, if you think about it, my strawberry probably had fungi and bacteria growing on the surface of it before I crushed it. So it's got the DNA of those in there too. So if we were to use this DNA in a strange cloning experiment, we would end up with some strange strawberry fungus bacterial hybrid. What a cool thought. Can you draw that? Thank you. Bye.